Welcome back, my dear students. In this session today, I am going to discuss the different gastrointestinal hormones. So let's begin our journey. What do we mean by the term gastrointestinal hormone? Gastrointestinal hormones includes all those specialized secretions from specialized glands or cells as well as the nerve endings related to the gastrointestinal tract. As we learn and move forward, we will come across different names of chemical messengers like gastrin, cholecystokinin, secretin, GIP, VIP, glycantin, glucagon, so on and so forth. All these chemical messengers, which are either produced by specialized cells in the gastrointestinal mucosa or related gastrointestinal organs like the pancreas, or may be produced by nerve endings that terminate on the gastrointestinal tract, are together known as gastrointestinal hormones. They, all of them, almost act predominantly by paracrine fashion. Even though they are being called hormones, which basically means a chemical secreted at one site and which will act far away from its point of secretion. Here, in the case of gastrointestinal hormones, that rule is seldom followed. Actually, most gastrointestinal hormones will act at sites near or just in the vicinity of their point of production. They hardly travel through the bloodstream and act in sites farther away from their point of production. So, paracrine fashion is the predominant fashion in which gastrointestinal hormones act. However, latest researches have indicated that these gastrointestinal hormones have counterparts that are being secreted in different parts of the brain also. There, their point of action is more in an endocrine fashion than in the paracrine fashion. So this demarcation line that gastrointestinal hormones will act by paracrine fashion is getting blurred with more and more knowledge we gain. Actually, it has been found that an entire part of the autonomic nervous system is dedicated to the control of the different gastrointestinal functions, both secretory as well as mechanical events. That part of the nervous system has been given the name as enteric nervous system, and the main executioner arms of these enteric nerves are these different chemical messengers or gastrointestinal hormones. So nowadays another term is being used to designate this nexus between the GI hormones and the enteric nervous system, and that is known as the brain-gut axis. When we will deal with nervous system, we will go into the details of brain-gut axis. Today we will skip to the discussion of the gastrointestinal hormones proper. So, what are the different gastrointestinal hormones? Broadly, they are classified into two families, namely the gastrin family and the secretin family, based on the structural homology of the different hormones. The gastrin family includes the gastrointestinal hormone gastrin and cholecystokinin. Earlier, the cholecystokinin was christened as cholecystokinin pancreozymin. Nowadays, the pancreozymin part has been removed and the name only cholecystokinin is given. The next family is the secretin family. That includes obviously the hormone secretin, then glycentin, GIP, which stands for glucose dependent insulinotropic peptide. Earlier the GIP was called as gastric inhibitory peptide. This name has now been changed to glucose dependent insulinotropic peptide. Next is VIP, which is vasoactive intestinal polypeptide, and glucagon. All these are members of the secretin family of gastrointestinal hormones. The first gastrointestinal hormone that I am going to discuss is gastrin. If you remember from my class of physiology of stomach, I had already given you a brief account of gastrin. It was the hormone that was secreted by the G cells located in the antral gastric glands. But other than that source, gastrin can also be produced by other gastrin secreting cells located in the duodenal mucosa, the islets of Langerhans of the pancreas, pituitary glands, and different parts of the brain concerned with mood and feeding behavior. So the predominant source of gastrin is obviously the G cells of the antral mucosa, but there are other sources. Structurally, gastrin is a linear peptide hormone. However, this is a simplified statement. It has been found that gastrin exhibits the phenomena of macroheterogeneity. That is, even though functionally all gastrin molecules have same stature, they are having variable lengths of their polypeptides. That is, same hormone having variable polypeptide lengths with preservation of its functionality. That phenomenon is known as macroheterogeneity, and that is exhibited by the GI hormone gastrin. It has been found that 3 different forms of gastrin are found in our body, namely big gastrin, little gastrin, and mini gastrin. Big gastrin has 34 amino acid residues, little gastrin has 17, and mini gastrin has 14. Even though functionally they are almost comparable, operationally the most potent gastrin is the little gastrin or G17 in terms of causing acid secretion. Other than macroheterogeneity, gastrin also exhibits another phenomena structurally, which is known as microheterogeneity. What is that? It is a difference in derivations of single amino acid residues of the different gastrin molecules. That is, the same amino acid at a particular position might have a sulfation in one gastrin molecule and an N-acetylation in another. So even though it is the amino acid which is same for that particular position in 2 gastrin molecules, one might having a sulfation and another has an N-acetylation. This type of difference in structure is known as microheterogeneity. So gastrin exhibits both macroheterogeneity as well as microheterogeneity. heterogeneity without much alteration in its function. So what are the different actions of gastrin? First of all, how does gastrin bring about those actions? You must remember from my class of stomach physiology that gastrin acted through the receptor known as CCKB. So the receptor for gastrin is the cholecystokinin B receptor. So even though it is the receptor for gastrin, the name of the receptor is CCKB receptor. And after gastrin binds to that CCKB receptor, lot of intracellular events take place, and ultimately it leads to the inositol trisphosphate and diacylglycerol, that is IP3-DAG formation and activation of protein kinase C, and through this pathway gastrin leads to its different cellular effects. Or in short, gastrin acts through the IP3-DAG and protein kinase C pathway. The details of these pathways you will not understand now, but when you will study the mechanism of hormone action as part of endocrine physiology, you will understand better. For now, just remember that gastrin acts through the IP3-DAG protein kinase C pathway. So what are the different actions? The first one you are already aware of. It increases gastric acid and Cassell's intrinsic factor secretion from the parietal cells and pepsinogen from the chief cells. The next action is also known to you. It increases gastric motility and thus helps in gastric emptying. The third action is it has trophic effects, that is, it promotes growth on gastric mucosa and stimulates parietal cell maturation. So basically, gastrin acts on the isthmic cells, and if you remember, those isthmic cells were mainly the stem cells. They could either become surface epithelial cells or become parietal cells or the chief cells. Gastrin promotes the parietal cell maturation and the flow of the isthmic cells towards formation of parietal cells rather than the surface epithelial cells. So it has trophic effects on the gastric mucosa and stimulates the parietal cell maturation. Another important function of gastrin which has been found is it prevents esophageal reflux of stomach contents because it increases the tone of the functional lower esophageal sphincter. So the stomach, when it is with food, will obviously try to give its contents into the duodenum. But if you remember, the gastric peristalsis occurs in two waves, a minor and a major ring. Since the minor ring is always closing the pyloric antrum before the major ring comes, so most gastric contents actually revert back into the stomach every time the major ring tries to push it into the duodenum. It is only the minor ring which pushes a small amount of chyme each time into the duodenum. Sometimes if the stomach is too full, that may cause reflux of food into the esophagus also. That is what leads to heartburns, and that is prevented by closing the lower esophageal sphincter, and gastrin is one of the mediators of that closure. Gastrin also induces insulin secretion, especially after a protein meal. Proteins means amino acids. Lot of amino acids are glucogenic amino acids. Some of them are ketogenic, but many of them are glucogenic. Once we devour them, once these amino acids are inside our system, they might cause a rise in blood sugar levels. So gastrin, by inducing insulin secretion, prevents sudden rise of blood sugar levels due to intake of a protein meal full of glucogenic amino acids. Next, what are the factors that stimulate production of gastrin? The main factors behind the production of gastrin, first is the products of protein digestion in the stomach, particularly amino acids like phenylalanine and tryptophan. And these amino acids can increase gastrin secretion directly by acting on the G cells. So first factor is the presence of products of protein digestion in the stomach. Next is vagal stimulation. Now, the neurotransmitter responsible for gastrin release via vagal stimulation is GRP, or gastrin releasing peptide, rather than acetylcholine. Even though in all other situations vagal stimulation means the neurotransmitter to be acetylcholine only, here the transmitter that is released by the vagal nerve endings for release of gastrin is different, and it is known as gastrin releasing peptide, or GRP. These act directly on the G cells to stimulate gastrin production. Also, stretch on the stomach wall with food material causes release of gastrin from the G cells, and increased calcium in the blood can cause gastrin release. So these are the factors responsible for stimulation of gastrin secretion. What are the inhibitory factors? Inhibitory factors, the first is a low gastric pH, means gastrin itself inhibits its production, because gastrin will cause increased acid production. Increased acid production will lower the pH, and once the pH goes low, it inhibits the G cells. So this is one example of a negative feedback in physiology, that gastrin itself causes its inhibition of secretion. Also, entry of acidic chyme in the duodenum stimulates a reflex known as enterogastric reflex, and this enterogastric reflex leads to release of various other paracrine mediators like somatostatin from the D cells, CCK, secretin, GIP, etc., all of which decreases gastrin secretion. So gastrin will be secreted when products of protein digestion are there in the stomach, mainly amino acids like phenylalanine and tryptophan, which are all glucogenic amino acids, and vagal stimulation, stomach wall stretch, and hypercalcemia, while gastrin secretion is inhibited by low gastric pH and entry of acidic chyme in the duodenum, which stimulates the enterogastric reflex and releases anti-gastrin hormones, namely somatostatin, CCK, secretin, and GIP. Lastly, Few applied aspects related to gastrin. These three are important, often asked in Viva. First is Zollinger-Ellison syndrome. Here there is hypergastrinemia, that is, there is increased gastrin level in blood due to production of gastrin by gastrin secreting tumors, mostly located in the pancreas, sometimes in the duodenum, and sometimes even in lymph nodes. Here gastrin levels are high and they act on the parietal cells to increase acid production. Also, these gastrin, which are all products of cells which are cancerous, so they cause watery diarrhea also, and there is lot of peptic ulceration due to increased acid production. Next is gastritis or chronic gastritis. Here also there is hypergastrinemia, just like in Zollinger-Ellison syndrome. But that hypergastrinemia is not due to any tumor that is producing excess gastrin. Rather, due to gastritis, there is destruction of the parietal cells. So acid production is low. As acid production is low, it leads to the phenomenon known as achlorhydria. So the inhibition of a low gastric pH on G cells is removed. Hence, the G cells are not under any inhibition anymore, so they produce more and more gastrin. Though this gastrin has nowhere to act in terms of the gastric parietal cells, because they have been destroyed by inflammatory disease or autoimmune diseases due to chronic gastritis. In these cases, gastrin acts as an important diagnostic marker. So a person suffering from symptoms like gastritis with high gastrin level, we can diagnose as a case of chronic atrophic gastritis. Similarly, there is a disease known as mucolipidosis type 4. It is a genetic disease, autosomal recessive in nature. The basic pathology, it is a lysosomal storage disorder. It is manifested as several neuropsychiatric problems, but the earliest manifestation of mucolipidosis is a gastritis and achlorhydria with high gastrin levels. Here also the cause of high gastrin is the achlorhydria only, but high gastrin level with gastritis, achlorhydria and some corneal cloudiness will give rise to the suggestion that that particular child is suffering from mucolipidosis type 4. So these are the three important applied aspects related to gastrin. All of them have high gastrin levels, but the cause of the high gastrin level is different in Zollinger-Ellison syndrome in comparison to gastritis or mucolipidosis type 4. The next GI hormone is cholecystokinin. The source of cholecystokinin is predominantly eye cells of duodenal and jejunal mucosa. But just like gastrin, it has now been found that different parts of brain and nerve of the enteric nervous system also produce cholecystokinin. As it is a member of the gastrin family, it shares many structural similarity with gastrin, and just like gastrin shows both macro as well as micro heterogeneity. The different CCKs found naturally are CCK58, that is CCK having 58 amino acids, CCK33, CCK22 and a very small CCK8. All these have CCK like activities, even though the amino acids number are different in all. What are the actions of CCK? Now this CCK, as you know from your knowledge of gastrin, can act through two receptors. The receptor for CCK are two, CCK-A and CCK-B. Gastrin shares the CCK-B receptor with CCK, but CCK has receptors CCK-A and CCK-B for itself. The CCK-A is mainly localized in the GI tract, while CCK-B is mainly localized in the brain and in the stomach. So these are the two receptors for CCK through which it acts. After binding to its receptor, how does CCK act? Just like gastrin, it acts by the IP3-DAG and protein kinase C pathway. And what are its principal actions? First of all, as its name suggests, cholecystokinin. That is, it causes contraction of the gallbladder and expels bile into the intestines, facilitating fat digestion. That is the most important action of CCK. Cholecystokinin, movement of the gallbladder, that contraction and ultimately expulsion of bile into the intestine. It also induces enzyme-rich secretion from the pancreas and thus helping in overall digestion of all proximate principles of food. That is the reason the name pancreozymin was also given to it, but nowadays the pancreozymin part has been removed from the name to add to the simplicity. What are the different proximate principles of food? Namely the three: carbohydrate, fat and protein. Pancreatic enzyme-rich secretion has enzymes to digest all three, and since CCK induces the secretion of all these enzymes, hence it is important for overall digestion of all proximate principles of food. Another action is it potentiates the action of secretin on the pancreas. What is the action of secretin on pancreas? I will come to later when I will discuss secretin. But one important action of CCK is it potentiates the action of secretin on pancreas. Obviously, you also know that as part of the enterogastric reflex, the CCK inhibits gastric motility, and another part of CCK is it stimulates small intestine and colonic motility. So basically it stops the stomach from giving something to the duodenum and helps in clearing of the small intestine and colonic materials. Next is stimulates the secretion of intestinal enterokinase. This is an important enzyme for activation of different proenzymes which are secreted from the pancreas enzyme-rich secretions. All these pancreatic enzymes are secreted as zymogens. They need to be activated inside the small intestine, and that is done by enterokinase. So the enterokinase is important because without it the enzymes will not be able to act because they will remain as proenzymes. CCK stimulates secretion of enterokinase, so it not only brings the enzyme-rich secretion into the small intestine from the pancreas, it also helps in its activation. It augments secretion of glucagon as well as insulin. So both insulin as well as glucagon secretion is augmented by CCK. Insulin secretion, if it is augmented, it will help because it will bring down the high blood glucose levels that accompany a meal. However, if someone is taking a meal which is very deficient of carbohydrates and has a lot of proteins which are mainly ketogenic, then insulin secretion can occur because of other factors related to food intake. Mainly gastrin can cause insulin secretion also. Stretch of the stomach wall, stretch on the duodenum also can cause insulin secretion. But in that person, the blood glucose levels will not rise high. But if insulin is high, it will put the blood glucose into the cells. So the person will have the propensity to suffer from hypoglycemia. That is prevented by the secretion of glucagon. The concomitant secretion of glucagon helps preventing a hypoglycemia induced by secretion of insulin. Next important action of CCK is it exerts growth effect or trophic effect on the pancreas. And another important action which has been found is it induces satiety, that is the feeling of satisfaction or fulfillment that follows a meal. Lastly, nowadays CCK has also been implicated to be anxiogenic and hallucinogenic. So these are the different actions of CCK. What are the factors that stimulate CCK production from the eye cells? The most important is contact of intestinal mucosa with products of protein and fat digestion, namely small peptides, amino acids and fatty acids. Among them, the most potent stimulator is fatty acids having more than 10 carbon atoms. Another important stimulator of CCK production is acetylcholine released by vagal fibers. Now, as the products of digestion enter the duodenum, they will stimulate CCK production from the eye cells, which leads to further production of more digested protein and fat. So this is an example of positive feedback mechanism in physiology. Gastrin production was an example of negative feedback. Here it is an example of positive feedback. CCK production. CCK will induce its own production and increase because more and more digested products of protein and fat are entering the duodenum. How does it stop? There is no known inhibitor of CCK release. Its secretion ceases when all food is completely digested. Once all food is completely digested and removed from the duodenum because of gastrointestinal movement, there will be no stimulation on the eye cells and CCK production will reduce or stop. The next gastrointestinal hormone is the secretin. It is a member of the secretin family. So it is not related to gastrin or CCK structurally. Actually, it is very important historically because it is the first hormone. Among all hormones, this is the first hormone to be discovered, and it was discovered by William Bayliss and Ernest Starling as early as 1902. They were experimenting on laboratory animals on the effect of denervation on pancreatic secretion following passage of chyme beyond the pyloric sphincter. So basically they denervated the pancreas and allowed chyme to pass beyond the pyloric sphincter and enter the duodenum. They found that the pancreatic secretions are still occurring, mainly the watery pancreatic secretions, and that is why they postulated that there must be a chemical messenger which is being secreted from somewhere which is causing this pancreatic secretion, and it has got nothing to do with nerves, and they named that particular chemical messenger as secretin, even though they didn't know that what is the actual nature of secretin at that time, but they postulated that there must be something which is causing the pancreatic secretion other than nerves, and this led to the concept of hormones and discovery of secretin. So what is the source of secretin? What do we know now? It is secreted by S cells, specialized cells located in the mucosa of duodenum and jejunum. Structurally, it is different from gastrin and cholecystokinin. Even though it is a linear peptide hormone, it does not show macro or micro heterogeneity. It is composed of 27 amino acids and bears structural similarity with other members of its groups, namely glucagon, glycine, GIP and VIP. Actions of secretin. Secretin, just like any other peptide hormone, will act through its receptor on the cell membrane. The receptor for secretin does not have any special name. It is just secretin receptor. And what is the nature of that receptor? It is coupled to a G protein. That particular G protein to which the receptor is coupled is activated when secretin binds to the receptor. Binding of secretin to the receptor activates the G protein, and that G protein activates further proteins downstream, namely adenylate cyclase. This adenylate cyclase enzyme causes cyclization of ATP to cyclic AMP. This cyclic AMP will further activate another protein known as protein kinase A, and this will ultimately give rise to all the actions related to secretin. So again, secretin binding to its receptor causes activation of a G protein to which the receptor is coupled. That G protein will cause activation of the enzyme adenylate cyclase inside the cell. The adenylate cyclase will convert ATP to cyclic AMP. An increased amount of cyclic AMP inside the cytosol will cause activation of protein kinase A and the subsequent actions of secretin. So this pathway is also known as adenylate cyclase, cyclic AMP, protein kinase A pathway. Now what are the main actions of secretin? As already stated, it is the hormone responsible for the watery bicarbonate rich secretion from the pancreas, and this watery secretion from the pancreas neutralizes the acidic pH of the chyme. This step is very important because if the pH of the chyme is not neutralized, the different pancreatic enzymes which act at a much higher pH than what is of the chyme will fail to act. So for optimal functioning of pancreatic enzymes, the watery bicarbonate rich secretion of the pancreas is essential, and it is brought about by the action of secretin. Another important action of secretin is it is part of the enterogastric reflex, just like CCK, and it inhibits gastric acid secretion and delays gastric emptying by increasing the pyloric sphincter tone. If you remember, CCK potentiated the action of secretin on pancreas. Similarly, secretin potentiates the action of CCK on pancreas. So both are going hand in hand. CCK will cause more release of watery and bicarbonate rich secretion from the pancreas due to the action of secretin. Similarly, secretin will cause more enzyme rich secretion from the pancreas due to the action of CCK. So both are synergistic in nature. Secretin also increases hepatic bile secretion. CCK causes bile secretion from the gallbladder, that is the stored bile, while secretin increases hepatic bile secretion directly from the liver, and that is why it is also known as a choleretic, that is a hormone or chemical that causes increased bile production and secretion. Now, other than these roles on the gastrointestinal tract, secretin has been found to play an important role in osmoregulation of our body, means the osmolality of our extracellular fluids. It has been found that secretin is a co-secreted agent along with antidiuretic hormone from the posterior pituitary. This antidiuretic hormone causes retention of water and reduced urine production. It is produced from the nerves of the posterior pituitary gland in response to a high osmolality of the extracellular fluids. Now, secretin has been found to be co-secreted along with ADH from the posterior pituitary, and it has been found that secretin receptors are present in the collecting ducts of the renal tubules, from where water reabsorption is increased. Now, secretin has also been implicated to be an anorectic hormone. That is, it reduces the urge to take in food and helps in energy homeostasis. So other than the roles on the gastrointestinal tract, secretin has roles in osmoregulation and energy homeostasis. So what are the factors that stimulate secretion of secretin? It is the acidic chyme and products of protein digestion entering the duodenum that will cause secretin secretion. And what is the inhibitory factor? It is the alkaline pH of small intestine. So once the small intestinal pH becomes alkaline, secretin production falls from the S cells. Now I will go to few other gastrointestinal hormones. The first one of them is GIP, or gastric inhibitory peptide, but nowadays known as glucose dependent insulinotropic peptide. So earlier name was gastric inhibitory peptide, but now named as glucose dependent insulinotropic peptide. It is secreted by K cells in the duodenal mucosa in response to presence of glucose and fatty acid in the duodenum. So glucose and fatty acid in the duodenum will cause secretion of GIP from the K cells. And what is its role? Its role is it is a part of the enterogastric reflex and thus decreases gastric acid secretion and motility. Along with it, it stimulates insulin secretion from the pancreas. That is why this name of glucose dependent insulinotropic peptide. The next gastrointestinal hormone is VIP, or vasoactive intestinal polypeptide. As its name suggests, it is both acting on blood vessels as well as the intestine. The action on intestinal smooth muscle is it relaxes the intestinal smooth muscles and it dilates the blood vessels of the gastrointestinal tract, mainly the blood vessels supplying the small gut. So The relaxation of the smooth muscles opens up the walls and the ducts of the glands, and the dilation of the blood vessels causes more secretion of watery substances into the intestinal lumen. So basically VIP causes copious watery intestinal secretions. It is secreted by the autonomic nerve endings innervating the gut, as well as a few specialized cells in the pancreatic islets, and it is secreted in response to acidic chyme entering the small gut. It is also a part of the enterogastric reflex along with secretin, cholecystokinin and GIP, and inhibits gastric acid secretion as well as motility. So along with its action of causing copious watery intestinal secretion, it is a inhibitor of gastric acid secretion and motility. Now, it inhibits the gastric acid secretion by antagonizing the action of gastrin, but it potentiates the action of acetylcholine on the salivary glands and has a co-transmitter like activity in different areas of the brain related to mood and food intake. So all these gastrointestinal hormones, if you appreciate, has some role in the central nervous system also, mainly in relation to our mood and food intake. VIP is no different. It also has co-transmitter like activity in different areas of the brain. So main action of VIP is obviously cause copious watery intestinal secretions, but other than that it causes inhibition of gastric acid secretion and potentiation of action of acetylcholine on salivary secretion. The next hormone we have already discussed in relation to stomach physiology is motilin. It was the hormone that was responsible for the migratory motor complexes during the unfed state in the stomach and duodenum. It is secreted by M cells present in the duodenal mucosa, and the main stimulator is a empty stomach or a stomach with a high pH, as well as a alkaline pH in the duodenum and jejunum. So all these cells which are producing motilin, they are stimulated in the unfed state and they are responsible for the migratory motor complexes, which are also known as the housekeeper contractions. There are few other gastrointestinal hormones whose names you will come across in textbooks or reference books, like peptide YY, which is again secreted from duodenal mucosa. It is a part of the enterogastric reflex and mainly it is a gastric inhibitory hormone. Also it has roles in food intake, mainly anorectic hormones, and acts as co-transmitters in different parts of the brain. Then you will come across names like ghrelin, bombesin, neurotensin, and so on and so forth. Actually, gastrointestinal hormones are a very labile part of your syllabus because new names are being added every year sometimes. So if you are interested, you can go to the library, you can go to the internet, you can search anywhere, you will find lot of names. You can add to your repertoire of gastrointestinal hormones, but I won't be discussing them in a general platform because they do not fall in the must know part of your syllabus. In your must know part, you have to know about gastrin, cholecystokinin, secreting. These are the must knows. And obviously few others like motilene, GIP and VIP. The others, if you want to learn, you can. If you feel that they are too much for you, you can omit. This is all about gastrointestinal hormones. So thank you for your patient listening and meet you again in the next class.